Hi, my name is Ida Osebe and I'm the curation team leader in EBRAINS. In this presentation, I would like to introduce the different options of data sharing that we have developed on our platform. But before I go into this, I want to briefly go into a little bit about open science and open data in general. So, open science means everything from open access journals and open and accessible peer review. It means open source software and tools. It means open educational resources, open and standardized methodology. And finally, and most importantly for this talk, it means open data. So, what is open data and why should we care? Getting information about data online can be massively confusing. Results are frequently overwhelming and unstructured, and they consist of mixes of articles, images, tables, and diagrams with a various use of terms that means the same thing. It also consists of different units of measurements and file formats. And this overload exists both for students as well as experienced researchers. And getting a thorough overview and screening takes a considerable amount of time. On the other hand, out of all research that is being conducted, only a small portion of it is available to the community. These are often interpretations of the results that are conveyed through articles, and the data itself frequently ends up on a dusty shelf somewhere. So currently, the possible links between the different researchers are blocked by obstacles such as paywalls and the necessity to get in personal contact with each other just to see if data is relevant. And this can be frustrating and ultimately slow down important research. So the most recent and maybe largest developments in open data is in open science is in open data. And this has recently been made possible by new technological capabilities to store large amounts of data and to work on and access this data over the internet. At high-speed internet and high computing power, the trends and developments of big data is to an increasing degree being applied to life sciences. So generally, what are the options for sharing data from your research? Several general data repositories for sharing research data are available. However, many lack the necessary stewardship and standards for making the data easy to find and use. If your dataset is large and or popular enough, you can also consider publishing your dataset in a in a standalone article in a so-called dataset journal. And Scientific Data and F1000 Research are examples of such data descriptor journals. So why are funders and global initiatives so concerned about the current state of science? These articles are examples of the combination of a large variety of studies that looks at the same topic or research question. These reveal a large degree of variability, which is often the case for these types of meta-studies. And the cause for this variability is frequently due to unknown and incomparable in-house standards. And variability often occurs alongside low reproducibility. And large variability between studies as well as low reproducibility can create a lack of trust in the results and makes it increasingly hard to reach an overall consensus for the neuroscientific community. And this lack of reproducibility is not a small-scale problem. Global initiatives are coming together because of trends that indicate that the current model status of many areas of science is not progressing as efficiently towards its goal, towards increases in scientific understanding, as would or should have been possible with improved methods and deeper knowledge. The current citation incentive could appear to more strongly favor competition rather than careful scientific inquiry and solid results. Unreliable findings are increasing, unreplicable findings are increasing, erroneous findings are increasing, all of which leads to retractions, which is what is reported on in this figure. And the number of years between each publication is increasing, as well as the time lag between the publication and retraction, which means that we are likely at an all-time high in terms of error and bias corrections. So science initiatives all over the world are beginning to tackle this issue by adopting more global standards and data sharing practices, such as for neuroscience and the International Brain Initiative, which was officially launched in 2018. The IBI has been established to coordinate efforts across existing and emerging national and regional brain initiatives. And a large portion of dealing with these issues is to start opening up data and making it transparent. But what is the optimal way to share data, and how would you go about to make it transparent? There are a lot of people talking about these FAIR principles, and you might have heard about them in detail before, but I still want to go through it because it's easy to understand what the words mean and less clear how they would apply to your own dataset and findings. So, making your data findable means that it's possible to search for and find your dataset without knowing about its specific existence in advance. So it needs to be tagged with descriptive metadata and persistent identifiers so that researchers that are interested in a particular trait can search for that trait only. And making something accessible means that something is retrievable in a relatively easy, global, and free manner, and paywalls are examples of limited accessibility. Interoperability refers to how the dataset's file formats, description, and structure is possible to use and combine with other datasets. So if you are, for example, using file formats that depend on non-common or costly software to run or open, then it has a limited interoperability. Finally, to be able to reuse the dataset, the necessary rights and licenses should be established in order to track the ownerships of the data, and we refer to this as provenance. So that was a little bit about the whys and the background to why data sharing is such an increasing phenomenon. In the next section, I want to highlight what we do and what EBRAINS is useful for. Because there are many data repositories out there, but there are equally large differences in how data is treated. And what we aim to achieve is increasingly fair data, and I will describe a little bit about how we achieve this on our platform. But for those of you who are unaware what EBRAINS is and represents, I want to give you a short introduction. The Human Brain Project has worked towards a concrete aim of establishing an e-infrastructure for neuroscientific research. And this is to ultimately benefit the individual researcher as well as the community. And this is an initiative represented by a large number of research groups in Europe. And as many of you are aware, EBRAINS is the place where the products that have been developed under the HPP are presented to the outside world. And we will be filling EBRAINS gradually over the final and coming phase of our European flagship funding. And a central part of this e-infrastructure is the collection, curation, and integration of research data. So the data that you submit from the various research groups across Europe is multimodal, heterogeneous, and differentially organized. All of these diverse datasets need to be categorized and described in a standardized way so they can be effectively searched, compared, and analyzed using integrated tools and services. And this is curation. Following all of the steps of curation makes it possible to link your dataset to relevant other datasets through a simple search. And in the backdrop of this search lies the EBRAINS knowledge graph and highly flexible metadata schemas that have been developed in the HPP, such as Open Minds. The datasets undergo spatial anchoring in our human and rodent coordinate systems that allow for visualization and regional searches based on anatomical location. And these features facilitate reuse and will in the EBRAINS e-infrastructure be coupled with modular analysis workflows that will enable researchers to analyze and model brain activity on large combinations of datasets. Before I go into more specifics about data sharing in EBRAINS, I want to briefly mention why data sharing and the quality of the service should be of interest to the neuroscientific researcher. Because it's not only to ensure that HPP and EBRAINS is getting its data for integration and reuse, and in that context, what would benefit other researchers, but what you personally or your group should keep in mind. So there are several clear advantages of sharing dataset from your research. And first and foremost, the currency of science today, citations. When sharing data on a data sharing platforms, that issues DOIs, you can be cited for all future work that uses your data. In addition, there's an increasing amount of research showing that the citations for the original paper belonging to the data also increases substantially. And this graph is from a recent paper showing citation advantages and displays the increasing trends towards open data when publishing in several journals. And this citation benefit is increasingly relevant if other researchers are able to search for and find your dataset in a trusted portal. Secondly, and also a second currency, is pleasing the funders. And open data and open science is an increasingly large focus area for many central funding institutions. And several are requiring a data management plan with an emphasis on fair data and fair and open data. And the increasingly applied fair data guidelines, as mentioned, indicate that data are as findable, accessible, interoperable, and reusable as possible when sharing data. And EBRAINS has had a focus on applying the same principles when building our data sharing platform and services. And in line with this, the curation service is built to enable data management to support for data that is published on EBRAINS, assisting with advice and tools to make your data more in line with the FAIR guidelines. These services also include long-term storage of your structured data in a federated and scalable data storage and computing infrastructure. And this also means a useful and secure backup of your dataset. Seeing as your work becomes increasingly visible and available to others, this can enable new collaborations without the need for personal contact in advance. And we welcome all types of neuroscientific data models and tools, and models were introduced last year and software was implemented 6 months ago. And here we have the EBRAINS platform and the entry point to its services. The EBRAINS knowledge graph search is where your dataset would be findable and accessible to the user. The platform uses metadata tags to sort and represent the data so you can more easily find what you're looking for. You can search by dataset, software, object, subject, and contributors. You can search by species. You can search by accessibility status, method, modality, or keyword. And here is a small video showing how it looks. So here you have a dataset card for a particular dataset. You have the metadata, the description of the datasets and projects, the people involved, the files, the DOI, a license, the publications connected, and a quick overview of the basic metadata for the entities. Also, EBRAINS datasets comes with a data descriptor that describes how to reuse the dataset in a concise and structured manner. And there are features to easily share the dataset with others. You also have the option to place your fully curated dataset under embargo, meaning that access is restricted to the data while waiting for instance, an article to be published. And it can be beneficial to complete the curation process in advance. For instance, if first author members of the project need to leave your research group before the article is finished. The EBRAINS search engine is built upon the EBRAINS knowledge graph. As curators, we add information as free text or append to existing entries. This creates connections between bits of information such as methods, types of subjects, authors, atlas coordinates, etc. And single datasets get integrated into a large system of interconnected nodes that builds up the EBRAINS knowledge graph. And this graph forms the backbone of the EBRAINS search. This graph system uses the Open Minds and Sans metadata standards and the underlying ontologies to standardize this graph structure. So if you want to prepare to adapt to these schemas, you can have a look at Open Minds and Sans on GitHub, and I will get back to these standards. All neuroscientific datasets share a feature. They are all localized somewhere in the brain. This means that they can be browsed and analyzed based on location. And our datasets undergo spatial anchoring in coordinate systems where they become linked to the EBRAINS Atlas viewers. We have a rodent and a human one. Here you can navigate and select a region in the 3D viewer. You can directly access and view the datasets from the knowledge graph search, and you can pin and save multiple datasets of your choosing, and you can navigate by region semantically. These interactive viewers are under continuous development and will include multiple features in the time to come. There are several options when you consider publishing your data on the EBRAINS platform. First, you can provide all forms of raw research data, models, and software that you consider valuable for an end user. For this, you submit a curation request. The request gets evaluated depending on our workload and the size and relevance of the dataset. Then the datasets undergo the steps of curation, where it's linked to the EBRAINS knowledge graph and appended to our Atlas coordinate systems. Finally, the data is published, where you receive a citable DOI along with a license, and after this your data can be reused, and it's optional to undergo more in-depth curation to more easily integrate to analysis workflows. But alternatively, you may choose to publish your dataset alongside a publication of your manuscript to a journal. And to an increasing degree, journals are requiring or recommending to provide the data of your publication to an appropriate repository. And EBRAINS is now a recommended repository for Nature scientific data. There are 3 options for this approach. You may want to publish ahead of your article and what is referred to as a preprint option. And following curation, you will immediately receive a DOI to append to your manuscript before submitting your article. The second, or the pre-published option, allows you to prepare your data for publication, release the metadata and description, but maintain an embargo or download restriction on the data until the article has been published. This enables the discoverability of your data and allows you to link to your dataset card URL in the EBRAINS knowledge graph. And upon publication of the article, we will release the embargo and you can append the DOI to your article. The third option is to initiate curation to prepare and finalize your dataset for release, but keep the dataset unreleased in its final version until the publication of your article is ready. And under option 2 and 3, reviewers of your manuscript will be provided private access to your dataset for reviewing purposes. And the steps of curation are key to optimizing your data sharing to adhere to the FAIR guiding principles. So I would like to describe the most important steps in our curation process. So the curation process is initiated by a request, and you can do so by going to the website or to contact us directly on curationsupport@ebrains.eu. Then a curator gets assigned to you. You deliver data and metadata. The basic curation takes place. The data sets get published and gets integrated with the atlases. And following a curation request, these are the most important steps for you as a researcher to complete. You fill out an ethics survey. You structure your data in an understandable and consistent way. You write a data descriptor, and this is a manual for users of your data, and having a rich and thorough data descriptor increases your chances of reuse. You fill out the Open Minds metadata form, and this fits your data to the metadata schema. And having a highly detailed metadata schema description leads to more people finding and using your data. You choose a license to share your data under, and finally you upload your data to our long-term storage. Any study normally incorporates a multitude of experimental research protocols, subject groups, methods, and in-house standards in how to organize data. And in order to effectively compare datasets between labs, a metadata standard is highly beneficial. And applying a common standard for all neuroscientific research has several benefits. Firstly, it involves a likelihood of error reduction, meaning that it minimizes misunderstandings, for instance, for data specifications. It involves a reduced need for curation, where standards makes it possible for researchers that were not directly involved in the research to understand and work on the data. And this means both in-house, between labs, as well as in public databases. It can lead to a optimized data analysis software, which makes it standardized and machine readable. And it can also involve the development of automated tools that allow for quick processing of big data. So, OpenMinds is a metadata standard that seeks to describe the whole span of neuroscience. And that is a challenge because of the broad dimensionality of the field. It is a high diversity of data formats, species used as model organisms, the size of areas, and the type of tissues targeted. Therefore, making this metadata standard and unifying this into one space is challenging. So the goal is for it to be flexible enough to capture experiment-specific features, yet strict enough to guarantee comparability across experimental data. So that is, OpenMinds must be fit to comprehensively describe both an electrophysiological rat study and human fMRI data. And at the same time, it must have a common denominators between these so that they can be compared. So finally, we have divided our curation service into a 3-tiered or layered process. And this has been created to adapt to the varying experiences, requirements, and engagements of researchers across Europe. So in our experience, researchers have very often have highly differential needs, and the level of engagement can be adapted to how fair you wish your data sharing to be. We undergo basic curation as tier 1, where the most essential information about your dataset gets registered. Then in tier 2, the datasets gets anchored via spatial coordinates in our Atlas integration process. And finally, in tier 3, you can opt to undergo in-depth curation, where datasets from certain domains, such as electrophysiology and calcium imaging, can be further curated and making them more readily integrated in combined analysis workflows. So this is what I have for you in this presentation. And if you are interested in hearing more about the curation service workflows and possibilities, please visit our website at eBRAINS.eu. So thank you for your attention, and I would like to refer to and thank the team behind all of the work that has been and will be done in our coming phase. And if you want to share your data, go to eBRAINS.eu.